Um, all right, hi everybody. Thanks for coming out to the first seminar of the semester. Also, thanks Dima for having me give the first seminar. I feel like this is a great way for me to kind of like uh, introduce myself to you all and kind of get a sense of like who's who a little bit. Um, before I start, just want to check in about whether everyone's got a neutral density filter. I've been handing out these little thin uh, filters. Everyone's got one? Cool. All right, great. So I'm going to use that for a little demo about halfway through my talk. Um, but uh, but yeah. My understanding of today's structure is that I'm gonna have three parts. I'm first gonna talk about myself, my journey, and then the main part of the talk is going to be the science. And then towards the end, I'll talk about future directions that I'm excited about. So um, so this is the title of my talk. I've tried to capture the work I do with this title. So it's quantitative characterization of human visual behavior, accommodation, and temporal processing in the visual system. So, um, wait a second, it's not okay. There we go. So I'll start by talking about myself. So I'm a new assistant professor here. I've been here for about 2 months now, and my area of expertise is pretty much the human visual system, various aspects of it. I run vision experiments for assessing vision. I do computational modeling of the resulting data, and I also image the optics of the eye, and perhaps that's why I'm here, at least part of the reason I'm here. Um, and as for, I guess, some non-work related stuff, here's a couple of things that I enjoy or am passionate about outside of work. So about my journey, I guess it starts in Malaysia, where I'm from. So we're this little country in the little red circle here. That's our flag. You can see that we actually have two little land masses that are separated by an ocean, which is kind of interesting. But we're well known for things like our food, our nature, and what used to be the tallest buildings in the world, like 20 years ago. Um, all right, so when I was in my teens, I moved to the US and I went to college at a place called Vassar College in Poughkeepsie, New York. That's about a 5 hour drive from here on the Hudson River. Um, Vassar's a pretty small school. That's most of the campus. It's a liberal arts college. And my original plan was to actually be a lawyer. I want to do pre-law. So, like these gentlemen here, who here knows this movie, by the way? All right, some of you. This is Philadelphia. There's Denzel Washington and Tom Hanks. Um, so that's my original plan because I like to talk about things I hyperfixate on. But problem was I lacked the verbal memory capacity. So I was taking a lot of classes in philosophy, English, and I was doing okay. But I was getting a little too many B's for my liking. So I decided to do a bit of a pivot uh, to cognitive science and a minor in computer science. I kind of have pretty broad academic interests, and so this felt like a good way to kind of maintain my all these different interests and reconcile them. And also because of two of my undergrad advisors whose classes I really enjoyed, and so they let me start working as a research assistant for them. And because this was a small school, there weren't any grad students, so I actually took on a lot of responsibilities. I wrote code for experiments. I ran the experiments. I also analyzed the data. Um, so I enjoyed it, and that kind of inspired me to go to grad school to do a PhD, which I did at the University of Pennsylvania in Philly. Uh, I did a PhD in psychology, working with Johannes Burge. I was there for 6.5 years, extended a little bit due to the pandemic. And then after I got my PhD, I drove across the country to UC Berkeley, the UC Berkeley School of Optometry, to work with Emily Cooper, an expert in virtual and augmented reality, as well as various aspects of vision science. Soon after I got there, a couple of months later, I acquired two co-advisors, Austin Borda and Marty Banks. And um, and so after 2.5 years there, I returned to the U.S. Sorry, to the East Coast by plane this time, and this is where I am now. So I was also told to include some lessons learned about from my journey. I don't know if I'm a good person to take advice from, but here's what I came up with. So do something you're good at first of all, because in the competitive economy, you want to have the best chance of standing out. You want to pick something you can enjoy at least a little bit, unless it takes a lot of money. How much money, that's kind of up to you to decide. But you want to have be consistently motivated. That's a big part of doing you know pretty much anything, right? Try to get along with people. Don't burn bridges, and be lucky. So, um, so a bit more about my research profile. So often in a lot of science areas, there's this like continuum. People often think of this continuum from being more theoretical to being more experimental. I've always seen myself as kind of being in the middle. I always try to balance the two. And then I have this other aspect which I picked up during my postdoc, which is imaging of the eye's optics. Um, as for how I think human vision fits into imaging science, this is what I think. I actually had these same slides in my job talk last year, but I really believe in this, so I'm going to say it again, which is that I see the human visual system as a great example, as a very remarkable example of an imaging system. It's shaped by evolution, but it lets us do a lot of remarkable things. And importantly, the visual system has basically all the important aspects of an imaging system, right? If you look at the eyeball, it's got a lens, right, with an autofocus feature, and that I'm going to talk about later. It's got an aperture, the pupil, and it's got an internal surface that's sensitive to light, just like a camera, right, or any sensor. Um, the analogy goes further though, because when you when you use your sensors or cameras and you collect data, you have to do some post processing, right? Same thing with the eyeball, right? Images captured by the eyeball have to go to the brain to be interpreted as aspects of our visual experience. Um, so on that, I'm about to start launching into the science part, but today I'm going to be talking about two projects that are both both kind of live in the visual system. They're projects that are largely complete, but have yet to be published. So I've actually not really talked about these projects here before. One's going to be about visual accommodation, which is the autofocus mechanism of the human eye. And the other one's going to be about depth perception and temporal processing. I decided to start with the latter first, because this is work I did in grad school. It's older work that I did with Johannes. We're still slowly polishing the project to eventually send it out to journals. We're both kind of perfectionists. Um, and so here's the title of that talk. It's called of this project. It's called Interocular Binding of Chromatic Signals Across Time. So why is time important in the visual system? Well, one reason time is important is the kind of fundamental fact that it processes different stimuli with different delays and latencies, right? Some stimuli are processed faster, some are processed slower. There's lots of good examples out there. For example, bright stimuli are processed more quickly than dark stimuli with less latency. Um, Low spatial frequencies are processed more quickly with less latency. If that's less intuitive to you, a more intuitive corollary of that is that blurrier images are processed with less latency. One way to think about that is that blur means there's less information, so it makes its way through the visual system faster. So this actually creates a problem for the visual system, which I and Johannes call the temporal binding problem, which is how does the visual system bind signals that have different delays and latencies, right? Because if you look at any visual scene, right, it's going to have many different stimulus properties. For example, you're going to have in a single scene like the one I'm showing here, you have both bright and dark. You have high contrast and low contrast. You have different colors, and different colors are also processed with different latencies, as I'm about to show you. And then even within the same object, right, you've got lots of local variations in the pattern. And so visual system has to bind these together, especially if the image moves. For example, if your eye moves or you move, right, your image in your eye is going to move. Um, and so the visual system has to have some way of kind of combining all these features. Otherwise, you perceive this image to kind of break up as it moved. But it kind of moves, we all see it moving rigidly, right? And so how is the visual system doing this? This is a pretty big question. And so I decided to answer a very specific instantiation of this question, where I'm only dealing with two different stimulus properties and asking how are they combined in time if they have different latencies. So here's one way to visualize this. I'm just showing you hypothetical visual system response as a function of time. And let's say you have a stimulus that it responds quickly to and a stimulus that it responds slowly to. So one way to put the problem is, should the visual system prioritize the faster signal to respond quicker, but in the process get less information? Or should it wait for all the information to come in? So it now has more information, but that's at the expense of quickness, right? So that's kind of the fundamental question. Of course, this is a very simplified diagram because we know that the visual system, just like any system, right? Its signals are not punctate in time; they're not little delta functions, but rather they evolve over time, right? They rise and fall, right? So what we're more likely dealing with is a situation like this, where we have two continuous signals that rise and fall, but one has a longer delay than the other. So how would the visual system combine them? Um, when I was trying to think through this problem, I decided to start as simple as possible. And often in science, the simplest possible model is a linear model, right? So I just thought, what happens if the visual system just adds the signals at every time point, right? Um, if it did so, under a lot of circumstances, you get something like this, where the combined signal has a delay or latency somewhere in between the delays of its constituents. So this creates a hypothesis that combining slow and fast signals, that combination is going to be more delayed than the fast signal alone. I'm not saying the visual system does this. This is my hypothesis. So with this hypothesis in mind, I needed a test case. And one test case I thought of that seemed quite appropriate was the cone photoreceptors in the eye. So the cone photoreceptors are little cells on the retina. So the retina is the little inner surface of the eye, the light sensitive inner surface. So we just zoom in on a little part of the retina. The cone photoreceptors are these cells here, the red, green, and blue, long things. They're called cones because their outer segment kind of looks like a cone. And what they do is they absorb light, turn it into the electrical signals that eventually make their way to the brain. The cones are what enables our very high resolution vision when it's bright outside, like in this room or outdoors. And they're also responsible for color vision. And they enable color vision because we actually have three different types of cones that are sensitive to different parts of the visible spectrum. From here on out, I'm going to call the long wavelength sensitive cones the L cones. I'm going to call the short wavelength sensitive cones the S cones, and I'm going to call the medium wavelength sensitive cones the M cones. And they're false color coded in this diagram because that's the color of those parts of the visible spectrum as we all know. So there's two reasons the cones I thought were a good test case. One is that the impulse response functions for the different cone types have been well measured by neuroscientists. So the temporal impulse response function just means how does the cone respond? How does its voltage change in response to a brief flash of light? And you can see here that the S cones, short wavelengths, these have the most delay, and the L cones, long wavelength sensitive, have the least delay. Right. So already we have two signals that have different temporal dynamics. Another reason to pick the cones is that there exists well-established methods for selectively modulating their activity. It's called silent substitution, and I'll try to explain that briefly. So this method of silent substitution kind of hinges on the fact that the spectral sensitivity profiles of each cone type have been well measured also, right? This is how much the cone responds to each wavelength for each of the three cone types. It turns out that if you want to know what the cone activations are for each cone type for a given light spectrum entering the eye, all you have to do is just take the dot product between the light spectrum and the spectral sensitivity curve of each cone type. It's a dot product, it's linear, right? This is kind of remarkable because very few things in biology are linear, but the cones, for some reason, are actually respond pretty linearly. So this actually enables this method we call silent substitution, it basically works like this. Let's say you've got a background of some light coming out of the screen going in the eye, and it produces a particular pattern of activation of the three cone types. Because everything's linear, you can just use matrix algebra to figure out how much do I need to change the light spectrum to just change the activity of the L cones in isolation. Okay, you can work this out just using matrix algebra. You can also do the same thing with S cones or M cones for that matter. But we're going to focus on the L and S cones today because those have the biggest differences between them. All right. But that's all we're doing. We're just going to wiggle the light spectrum and selectively modulate just one cone type. All right. So just to take stock of what I've told you here, right? I've told you about the fact that the cones have different, that the L and S cones have different delays, and we can selectively modulate their activity. So now I got interested in this again because of this question of what happens when you combine a fast signal and a slow signal. So now we have a fast signal L cone, and a slow signal, the S cone. And now I've actually colored them so they approximately resemble what the colors of these cone activations actually look like to an approximation. Um, and so again, my hypothesis is that the combination will be slower, more delayed than the fast signal alone. All right, so at this point now I'm actually going to start talking about I'm going to start foreshadowing the experiment I decided to run. So what I decided was that, okay, if I'm predicting that combining an L and S cone modulation is slower than the L cone modulation alone, I should be able to uh, run an experiment where I present a combined L plus S cone stimulus to one eye and present an L alone stimulus to the other eye. The reason I decided on this specific configuration is that there's actually a behavioral signature that you'd see if the processing in one eye was slower than the other eye. That's just a classic visual illusion known as the Pulfrich effect. So that's what. So here's where the neutral density filters I handed out come in play. So what I want people to do is kind of look at this stimulus, but only cover one eye with the neutral density filter. So make sure that one eye is completely covered by a neutral density filter, and the other eye is completely uncovered. And I'm going to ask if any of you see the stimulus kind of moving in an elliptical pattern a little bit. Does anyone kind of see that? So it might be subtle because it depends on how well the projector is synced up. But like for some of you, it might look like this bar is in front of the screen sometimes and behind the screen at other times. Is that working for some people? Let's not show of hands who sees this. All right, so at least some people see it, right? Actually, that's a pretty good number. Given that this is a projector, I haven't tried to parameterize anything here, right? So why is this illusion happening? I'll try to explain. And this illusion is about delay in processing between the eyes. So here's my attempt to, and we'll try this again. We'll come back to this in a couple slides. But what's going on here? I'm going to try to explain this. So here's a top-down view of a pair of eyeballs. We're looking down on the pair of eyeballs, and then here's the screen. The line is the screen. The stimulus is a little dot here. And I'm going to make this a GIF of what's going on, which is as follows. So the white dot on the line, that's the stimulus on the screen, right? So it's just moving left and right. It projects it to each eye. The image it forms in the right eye is this little white dot here. And the image it forms in the left eye is also the white dot. But the gray dot here is meant to represent the effective image. And it's gray because it's the color of the neutral density filter that you just put in front of your eye. And the idea here is that darker stimuli are processed more slowly by the visual system. So it's like your sensors are delayed. Okay. So the gray image here, the darkened image, is going to lag behind the actual position of the image as it moves. You don't have the problem in the right eye because there's no neutral density filter covering one eye. So what this creates is a discrepancy in the position of the effective image between the eyes, what is called in vision science a disparity, a binocular disparity, which the visual system interprets as an object in the world that's at a particular distance. And that particular distance happens to be not at the distance of the screen, but behind or in front of the screen. So the upshot here is delaying the stimulus in one eye by darkening it creates this illusion of depth. And you can actually change the direction of this effect if you just change which eye gets which filter. So just back up to the slide. So those of you who are able to see the effect, you can try covering the left eye and then covering the right eye. And if you do that, you should see the direction of the elliptical trajectory reverse depending on which eye is getting which one. Gabe.
Good question for you. So we're having this, you're changing our subjective experience. Does it also change behavior? So have people used eye trackers to find that people divergent eye movements, the binocular eye movements are different?
That's a good point. I'm not aware of any studies trying to use eye trackers to explore this illusion. If you're talking about like effects on behavior, so Johannes, my advisor, actually got really interested in the Pulfrich effect, and the second part of his like pre-tenure period. Yeah. And one thing one pitch he's often made about the Pulfrich effect is that it could affect driving behavior, because there's a version of the Pulfrich effect that can happen when you have instead of different light levels between the eyes, different amounts of blur between the eyes, and that's what bifocals do, right? some people with presbyopia correction, right? One eye is corrected for far, one's corrected for near, so they can both read and look far away. But that introduces a blur between the eyes. And so Johannes's kind of pitch there was that like that could impact driving behavior because if you have to brake suddenly and you're misperceiving the depth of the pedestrian, it could potentially have fatal consequences. So like as far as I know, he's still running like large scale studies to try to figure out how much this really would have an impact in a situation like that.
Yeah.
But you asked about like kind of behavioral consequence, and this is something that like I know Johannes is exploring.
I also wonder, so how about we could have an interesting conversation right now, but I'm going to sidetrack you. So cool, awesome, thank you.
Cool. All right, yeah. Let's let me talk more about this.
Gabe cutting himself off.
Yeah. Yes. Uh, I'd say this is probably happening farther up in the brain. So I don't think the photoreceptors at the eye level it's really this delayed. I think the evidence points to it's somewhere in visual cortex.
Yeah.
Right. So that's actually that's a very reasonable like a hypothesis. But the interesting thing about the blue light sensitive cones though is that they're actually slower. The responses are actually slower than the other two types of cones. The kind of like photochemical properties.
Generally, is there blue light's not as important?
Well, I don't know because I think the S cones and so the color people could correct me here. I think the short wavelength sensitive cones actually evolved earlier than at least one of the other two cone types. So it's a pretty evolutionary old system.
I had to make an uneducated guess as to why it might be longer integration times because we have fewer blue cones on the retina.
Oh, so you need to gather
have
more,
integrate longer in order to
gather more information.
Sampling the visual.
Yeah,
See, so
that could well. That's a
just wild guess.
Yeah. This sounds reasonable to me though. I mean, hard to test, but I mean, logically pretty consistent. Cool. So this is the classic Pulfrich effect. That's been known about for a long time. But you remember I was talking about I spent all the time talking to you about L cones and S cones. And so we want to actually leverage that knowledge to see if we can produce a Pulfrich effect without changing the luminance of the brightness between the eyes, but instead if we just change the hue and saturation between the eyes, right? So in one eye you'd have L plus S modulation. I guess I already told you this. Combined L plus S modulation in one eye and L alone the other eye. And this would actually, under most theories of color, not change luminance because it's thought by many in the color science community that the S cones contribute minimally to our perception of brightness. Again, color people in the audience can correct me if there's been newer knowledge on this front. But yeah, if you're able to show this would be quite cool because as far as I know, this has not been shown before that you can get the same effect with just a hue difference between the eyes. All right, so that's the game we played. One color to one eye and a different color to the other eye. To do this, we use an apparatus known as a haploscope, which consists of two monitors and some mirrors to read. So this would be the front view of the haploscope. So the subjects they'll put their chin here where it says chin rest. Start using laser pointer, and their eyes would kind of be aligned with these slots. And then here's a top-down view of a haploscope, right? So there's monitors, and you can see that there's a system of mirrors that ensure that each eye only sees one monitor. Okay, so you're really presenting a different stimulus to each eye. So the game plan here is different color in each monitor, in each eye. Move them, just like I moved that stimulus you saw earlier, and then see if you could get a similar effect. So this is kind of what it would look like. Here I'm showing the left eye image and the right eye image, different colors in each eye. They're oscillating back and forth. The subject's task is to report the direction of the depth. So whether it's basically moving like this, if you follow my finger, or moving like that. And I've represented here. So here's a top-down view of the trajectories. Either it's moving left when it's in front of the screen, or it's moving right when it's in front of the screen. I can't demonstrate this in the auditorium, obviously, because I don't, I can't literally present a different image to each eye for you all. And these color differences also wouldn't, I'd have to calibrate this projector to really get it to work. But anyway, that's the key manipulation. Change the color between the eyes. Another manipulation I made is to introduce on-screen delay between the eyes. So here you can see the magenta. So the L plus S stimulus is the magenta looking one. The beigeish pink one is the L. You can see that the magenta looking one I made it on screen. It's now leading the pinkish one, the pinkish beige one, right? Um, and this is just to create to simulate some actual motion and depth trajectories for the subject. My goal here was to just evaluate to see whether this Pulfrich effect would show up for different kind of base paths that the stimulus is taking. Okay, so here's the data. So here's this data takes the form of a psychometric function. Some of you have, I guess, hopefully a good number of you have probably taken the human vision and psychophysics class, right? So this is a psychometric function, if you remember that. It shows you how often the subject reported this type of trajectory as a function of the amount of delay on the screen. And so if you didn't take the class or you don't remember it, that's okay. Because the thing I really want to draw people's attention to is the fact is the difference between these two psychometric functions that I measured. Okay, you can see that they're clearly shifted, right? At every on-screen delay value, right? One function is systematically higher than the other. And the only thing I did in the experiment to produce these two patterns of data was to switch which eye received which color. Okay, so the color of the curve indicates the color that was in the left eye, right? So all I did is I swapped the colors between the eyes, and these are interspersed. These trials are interspersed, so it's randomly. So the subject, for the record, the subject actually doesn't like they see. Just to be clear, the subject sees one stimulus. I'm presenting a different stimulus to both eyes, but both eyes combine uh, the images between the eyes into one single percept. Okay, so the subject doesn't know what color is going in which eye, right? But we can get this very systematic shift in the psychometric function. I guess I'm just going to pull the room because I'm teaching this class right now. So the 50% point, the on-screen delay that gives you the 50% performance value, what's it called? Does anyone remember? Let's shout it out if you want to. It's the point of subjective equality, or the PSE. Okay, so there's a very systematic shift in the PSE, right? This is just one condition from one subject. Here's all the rest of the conditions, and the differences between the plots are that as we go from left to right, I'm increasing the amount of S-cone contrast I add to create the combined L plus S stimulus, right? So I'm adding more of the slow thing, right? S-cones are slower. I'm just increasing the amount of S-cone I add, and as I do that, you can see that you get a very systematic. This shift is not only systematic, but it grows bigger. And it turns out that under some fairly reasonable assumptions, if you take this shift in PSE, the point of subjective equality, and divide it by 2, that should give you the delay caused by the color difference. Okay, so we can summarize all this data in a plot like this, where I'm just showing the estimated delay and how it changes as a function of how much S-cone contrast I add. And this is for one subject. This is for the other subjects I tested, and this is the average, right? It's, pretty remarkably linear. So the conclusion here is that per my hypothesis, when you combine a fast thing in L and a slow thing in S, the combined stimulus is slower than the fast thing alone, and it grows. It slows down the more of the slow thing you add. So this is just a kind of visualization, revisualization of my hypothesis, right? Sorry, I combined a fast thing and slow thing that has more delay than the fast thing alone. And along the way, we've also recreated this Pulfrich effect, but in a different way that to my knowledge has not been shown before, where you're creating this illusion of depth with just hue and saturation differences, not luminance. And so I have some future directions in mind for this project, but it's actually not my focus right now at RIT. The second project I'm going to talk about today is the one that's more directly related to my plants. Jim.
Is rivalry a problem at all?
So I think there are some. When I was piloting this experiment, you saw how I introduced on-screen delay. There are certain points when I was still piloting the on-screen delay where I would have so much on-screen delay that I would start to see this. I would no longer see a single stimulus, but it would kind of split into two. But I realized you were asking about rivalry, color rivalry. So these stimuli, that's a good question, actually. So these stimuli, when you fuse them, they actually look a little bit shimmery. Because I mean, as you know, right? That's what happens when you have binocular rivalry. So yes, you do see some shimmer. I didn't systematically ask my subjects about that, but I know when I do this, I definitely see a kind of shimmery um uh fused stimulus. Do people have any other questions about this part of the talk? They're going once, going twice. All right, so let me just see how I'm doing on time. Okay, it looks pretty good right now. Might be a little bit ahead of schedule actually. Um, all right, so that was my a project I did towards the end of grad school. Now I'm going to talk about the main project I've worked on during my postdoc with my three advisors at UC Berkeley. Although I mainly worked with Emily. Um, so this is on visual accommodation, which is the autofocus mechanism of the lens in the eyeball. And the title of this project is Determining the Wavelength and Focus During Accommodation to Polychromatic Stimuli. So I'm going to unpack that. But first, I want to talk about accommodation, right? So one goal of the visual system is to obtain a clear image, right? Many visual tasks require a clear image. Probably most visual tasks require a clear image, right? For example, reading. Accommodation is how the visual system obtains and maintains a clear image on the retina, right? It's the changing of the power of the lens in the eye using little muscles in your eyeball to produce a clear image. This is a very important oculomotor function, and we do this thousands of times a day, at least up to middle age, when it gets harder to do, as some of you might know. Um, and so right here, I'm showing a situation of accurate accommodation where a point in the world uh, is focused just the right amount, so it shows up as a point image on the retina, right? And so that's how you get the sharp image. Under accommodation happens if you have too little power in your lens, and so now the point is instead focused behind the retina, and that causes blur. You get the blurry image. You could also instead be over accommodating, so you have too much power in your lens. So now the image is formed in front of the retina, and that also shows up as blur um, visually, right? So now next question is, how does the wavelength of light affect blur and accommodation? What does that have to do with things? So uh, uh, we can appreciate this from the simple fact that natural scenes are polychromatic and broadband. This is an audience of imaging scientists, many of whom do remote sensing, right? So I really don't need to impress this upon you, right? Like light from the natural environment is broadband. Its energy is spread across a range of wavelengths, right? Even for things with distinct colors like blue flowers, right? Still very broadband. And so this matters for the visual system because this broadband light interacts with chromatic aberration in the lens. I'm going to focus on longitudinal chromatic aberration, or LCA, which is the fact, this refers to the fact that different wavelengths of light are refracted by different amounts as they pass through the cornea, the front surface of the eye, and the lens. And the consequence of this is that only one wavelength can be in focus on the retina at a time, even though you've got lots of wavelengths coming into the eye. So what this does practically is that this creates little color fringes on the retina, right? Here I've got an example of green being in focus. So now you've got like a little blue fringe around, sorry, purple fringe around it, right? Because red and blue are out of focus. Um, and but the specific pattern of color fringe depends on the wavelength and focus. So here I'm showing you examples of long wavelengths in focus, middle wavelengths in focus, short wavelengths in focus, right? And you can see the color fringe pattern varies systematically. Um, so, okay, and here I'm just kind of zooming in on the images a little bit so you can see what I'm talking about, right? This color fringing pattern that depends on which wavelengths in focus. So what does this mean for accommodation? Well, a classic theory in the vision science literature is that this actually doesn't mean anything for the visual system. Like this classic theory says that the visual system is just going to convert these color fringes to grayscale, right? And then just try to maximize the contrast of this grayscale. So it's going to just use the luminance mechanism and maximize the contrast or image quality of that. This view was later on, and by maximize, just to clarify, I mean changing the idea here is that it just the visual system just kind of in real time changes the focal power of the lens until the image looks as sharp as possible, just trying to optimize the sharpness using some kind of search. But this theory is later contradicted by some classic work from at least two labs showing that actually, depending on what color fringe pattern you have on the retina, on the retina, that actually will stimulate the visual system to accommodate in predictable ways. Namely, if long wavelengths are in best focus, the visual system tends to kind of automatically decrease lens power, and it'll do the opposite if short wavelengths are in focus, right? And the theory here is that the visual systems are actually trying to get green into focus, so the middle wavelengths. Those are the ones that appear brightest to us, right? So it's just using the pattern of color fringing to decide, okay, am I focusing too much or too little for green? That's the theory here, right? Now some pretty recent work actually from Jenny Reed's lab at Newcastle has kind of done a variation of this work where they present stimuli that are made up of only long or short wavelengths, and they vary the relative intensities of these different wavelengths. And they showed that when they do that, they can actually push accommodation around pretty predictably. So this is recent work that for my postdoc project I wanted to build upon. Because one thing about this work is that they showed how the refractive power of the lens changed with these different mixtures of wavelengths. But they didn't actually report the wavelength in focus because that's a little bit more involved. And so the goal of my study was to kind of build on that work and try to directly report the wavelength in focus using similar stimuli, varying the ratios of long and short wavelengths, and in doing so, use this as an opportunity to try to identify the mechanisms, the visual or brain mechanisms driving accommodation. And this is a potentially useful endeavor because it could be useful for things like evaluating image quality, or even the mechanisms of myopia. Myopia is short-sightedness. So the development of short-sightedness. So we set out to do this, but first I have to tell you how we estimate the wavelength in focus. Okay. So here's the apparatus we use to present our stimuli. Okay, so we have an OLED display with narrowband primaries. So primaries are just the pixels. These are the spectral emission profiles of the RGB pixels. And then we have a focus tunable lens. This is a liquid lens that can rapidly change its focal power in under like 10 milliseconds. And we could use this to simulate the display being at different distances. And we also designed the apparatus to minimize magnification. So no matter what distance the display is at, it appears the same size. There's an optical relay, which I won't talk about in this context. It's not that relevant to my project. So our subjects, we put an eye patch in the left eye. They're only viewing it with the right eye to kind of try to isolate the accommodation and not bring in any binocular mechanisms that involve both eyes. And then we stabilize their head with a bite bar, which is a piece of dental acrylic, which they bite on while they're doing the experiment, so that we can measure their accommodation as best as possible. Their head and eyes won't move around too much. What we use to measure their accommodation is a wavefront sensor, specifically a Shack-Hartman wavefront sensor. That's a principle that it uses. It's recording 875 nanometers. Basically, what this does is it captures all of the optical aberrations of a particular person's eye. And from that, you can work out things like the point spread function. So here's a real example point spread function that we measured from a subject. It might be mine actually. I'm not sure. And so this is our apparatus. The task was very simple. Okay, so in this experiment, what we had people do, my subjects do, was that the stimulus would appear. It's just a 3-letter word pattern confined to 1 degree across, so it's pretty small on the black background. And then the subject's instructions are just get this into focus, and then when it looks in focus to you, hit the button. And when they hit the button, that's going to initiate 3 seconds of recording their wavefront, and then the next stimulus appears. So just to make the task a little bit more interesting, I had them do various permutations of these 3 letters. Still not that interesting, but a bit of variety doesn't hurt. But the important manipulation we made here was actually the stimulus color. So this table here illustrates our different color conditions. Each cell corresponds to a color. Each little box within the cell represents 1 unit of luminance for the red, green, and blue display primaries, respectively, pixels, right? And so you can see here, as we go from left to right, our stimuli run from the gamut from having more long wavelengths to having more short wavelengths. And the top versus bottom row is just the presence or absence of middle wavelengths. And then we also presented these stimuli at 3 distances. Here I'm showing them in diopters. which is roughly 67 centimeters to 29 centimeters. And we had 6 repeats per combination of color and distance. So here's some actual traces, continuous combination traces that we recorded from the wavefront sensor. So on the y-axis, this is defocus aberration at the wavelength of at the recording wavelength of 875 nanometers. Defocus aberration is the aberration that changes the most with accommodation. And this is a time series because we're recording at 30 hertz. And so you can see, even though the data is kind of noisy, right? You can see there's a clear difference between these two color conditions I picked out, right? So this one has more short wavelengths. This one has more long wavelengths. Like despite the noisiness, you can see there's a pretty clear difference in means, right? And so what we do is we're in the next couple of slides, when I talk about this data, I'm going to talk about the average data, which is represented by this line here. Okay.
Something in these, that's the something that's happening.
That's a really good question. There are various theories for why this is happening. Some of it is probably noise, but the ones that are within like the 3 to 5 hertz range are often called micro fluctuations. And these basically happen anytime you're trying to hold something steady. So the theory is that at least some of these micro fluctuations are due just to like your pulse.
What's the response time of the lens to muscle?
Like it could be that I'm not really an expert on the biomechanics of the lens, but it could be a little bit of that, right? But yeah, maybe the interaction. Right. So maybe that's that kind of tracks, like, the range of frequencies I said. But yeah, there are some people who think that it could be this hunting for the best focus. Although it's actually kind of hard to run studies to test that hypothesis. So I think the evidence on that is pretty it's still pretty there's no consensus on that. But yeah, so this is the raw data. But for most of my analyses, I'm going to average across the entire 3-second period, as well as the across trials.
Did you look at people that have severe astigmatism?
So I wasn't too picky about my subjects. I had one subject who had really high astigmatism who actually couldn't pass the screening test for this experiment. So that's a whole other story. I had to screen some participants because the accommodation in this kind of scenario where you're only using one eye and it's dark is actually, like, some people have a lot of trouble with it. So I had to screen out some subjects, but one subject got excluded because they had really high astigmatism. But for the rest, I think the highest I saw was maybe like 0.75 cylinder. So, moderate astigmatism. That's about what I have in my right eye. Um, so yeah, not too much spread. Subjects who are myopic, we just put contact lenses on them because the wavefront sensor can also capture, the combined aberrations. Um, so anyway, now I'm going to talk about the process of determining the wavelength and focus, which first of all requires a model of the chromatic aberration, the LCA. Specifically, we need a model that can tell you the difference in the focus between any two wavelengths, right? But if you have such a model, that gives you a lot to work with, because now all you have to do is measure defocus at just one wavelength, right? In this case, the wavelength of our measurement device. And then now the defocus at all other wavelengths. And then just, simple fact of optics in this case is that if you know the distance of your stimulus, let's say you have stimulus of X diopters, then the wavelength with a defocus of negative X diopters is the one in focus, right? So in this example, X is 2.5. So we just look up in this graph, what wavelength does negative 2.5 correspond to? And that's the wavelength in focus. To constrain our estimates of wavelength and focus, we actually tried to estimate this function individually for different subjects. I don't have time in the main part of the talk to discuss the method we used for that. But I'm happy to talk about it at the end. It was a behavioral method for trying to estimate their chromatic aberration functions. And we used a task that was already established in the literature, and then the curves themselves are a parameterized fit using a model from this paper that's kind of physics inspired. It considers the fact that the lens and cornea are mostly water, right? So the refractive index, you can kind of build a model of chromatic aberration. But yeah, now here's to the main result: wavelength and focus, and how it changes. So it's on the y-axis. I'm going to show you how it changes as a function of the red-blue ratio. Okay. By red-blue ratio, I mean the ratio of luminance of the red pixels and the blue pixels. And on the plot here, I've also marked out the peak wavelength of the red, green, and blue pixels. So, and I've also marked out the graphic representation of the colors on top. But yeah, here's the first set of data. Okay. So I split the plots also into conditions with no green and conditions with some green. If you recall the set of conditions I showed you at the beginning. But what this is showing here is that for stimuli at a distance of 1.5 diopters, right, you get this very systematic shift in the wavelength and focus as you go from having, in this case, 4 times as much blue to having 4 times as much red. So this is consistent with previous work showing that humans accommodate more when you have more long wavelengths in the stimulus. But we're trying to directly report the wavelength and focus. We also find that as the stimulus gets closer, so going from 1.5 diopters to 3.5 diopters, the wavelength and focus also shifts shorter. And that's consistent with previously reported biases in the literature, that just biases as a function of distance. But the kind of interesting thing I like to emphasize about this data is the fact that the wavelength of focus is only about shift is only about 40 to 60 nanometers, despite the fact that you're going from having 4 times as much blue to having 4 times as much red. And these, if you recall, these arrows indicate the peak wavelengths of the different pixels, right? So between blue and red, that's about 160 nanometers of difference in their peaks. But our subjects only shift by about 40 to 60 nanometers. So our next question, kind of more kind of conceptual question, is why would the visual system shift the wavelength and focus by the observed amount, right? Why so small, right? It's very consistent, but it's a relatively small shift. And so I came up with a hypothesis for why this might be going on in the visual system, which is that it might be this behavior of the visual system might be its attempt to maximize image quality. And so first of all, this raises the question of how do we operationalize image quality? And so here's how. So to do that, I needed a forward model of the visual system. And here's how it works. So this is an example stimulus, and it's spectra. What I do is I first pick a wavelength and focus, and then I pass the stimulus through an optical modeling toolbox called ISET-bio that models the various optical aberrations of the eye, and also the cone photoreceptor responses. Remember, I talked about the cone photoreceptors in the previous section of this talk. And out of the ISET-bio, it spits out the image, the image on the retina, as it would be encoded by each of the 3 photoreceptor types. Now, it's generally accepted that in vision science that if you combine the outputs of the L and M cones, you get luminance, which closely tracks brightness, basically grayscale, right? So what I'm doing is I'm pushing through the stimulus through this optical modeling, and then I'm getting the grayscale image that is encoded by the visual system. And then now what is image quality in this case? How I operationalized it was how similar is this grayscale image in the visual system to the original image, right? So to get the similarity, the simplest possible measure I could think of is just to do a normalized cross correlation and get the peak cross correlation, right? This entire loop is for a particular wavelength that I picked to be in focus, right? But I need to repeat this for different wavelengths in focus, and this plot here shows how the image quality, which we call a peak correlation, changes as a function of the wavelength in focus. And the prediction of this model is that the visual system is going to pick the wavelength of focus that maximizes image quality. All right. We also did some bandpass filtering, which is not that important for the purpose of this talk. I'm happy to talk about it offline. There are only a few free parameters. One was the ratio of the weights on the L and M cones to account for individual differences between subjects. And we also had 2 more parameters just to account for these distance-dependent biases that you saw in the data earlier, right? So we fit this model to the data, and you can see that it qualitatively recreates a pattern of data, right? You have more long wavelengths, you shift your wavelength focus longer, but the model you can see is too aggressive, right? It predicts too large a shift in the wavelength and focus. And so we asked what is missing, right? What might be missing from this model? And so we went back to the drawing board and re-examined how we're combining the cone photoreceptor outputs. Specifically, we re-examined this idea of adding the L and M cones to get luminance. Basically re-examining our assumption that the visual system is trying to maximize image quality in the luminance channel. The reason we did this is because of some studies that I cited earlier in this talk that suggest that the accommodation might actually be driven by those color fringes. Remember the color fringes, right? Uh, Basically, those amount to differences in blur across different wavelengths. That's what the color fringes are. And so, and this previous work suggests that the visual system is picking up on these differences between the color fringes to drive accommodation, right? So now we're doing a comparison, right? We're comparing blur across wavelengths, right? That invokes the notion of a difference, subtraction, right? And so a mechanism that just has addition in it, right, can't really do this comparison I'm talking about. If on the other hand, you now throw a minus sign in there, right? So now we're subtracting M cones activity from L cone activity, right? Now that's a plausible way you could be doing this kind of comparison, this difference computation. And in the color science world, that's known as a red-green opponent channel. Now I'll talk about this more in just a second, because there's another type of opponent mechanism where now you involve the S cones, and now you'd be subtracting their outputs from the sum of the L and M cones, and that's called a blue-yellow opponent mechanism in the color literature. If this phrasing like red-green opponency, blue-yellow opponency sounds kind of foreign to you, right? Kind of one intuition there is that that when you think of a color, right, a single color, it's really hard to think of a single color that's both red and green, right? You could have objects that have red and green in them, but a single color that's both red and green, that's very hard to imagine. Same thing with blue. It's really hard to imagine a single color that's both blue and yellow, right? So these are thought to be kind of like fundamental dimensions of color in the visual system, right? That's why we can't reconcile them. So long story short, we decided to rerun this model, but now we're maximizing image quality in these color opponent mechanisms. Okay, and we get much better fits to the data, right? So here's a model that maximizes image quality in the red-green opponent channel. You can see it still underpredicts the data a little bit, right? But it turns out that if we maximize image quality in the blue-yellow channel, we get a noticeably better fit to the data. And so this is kind of the model that won out. And one way to look at it is that the blue-yellow model was the best fit for 5 subjects, 5 out of our 8 subjects. The red-green was the best fit for the other 3. But the key thing is that the model that maximizes luminance does not fit any subject better than one of the color opponent channels. So we decided to go with the blue-yellow model because based on these analyses, it was the best model. And so that's basically the takeaway from that's one of the takeaways from this talk, right? Well, there's 2 takeaways from this talk. One is that the wavelength and focus very systematically with a relative proportion of long and short wavelengths, and that this behavior is quantitatively consistent with a mechanism that maximizes image quality, or I guess you could also call it some form of sharpness in the blue-yellow channel. If you're curious about intuition for why the model is able to, yeah.
So this means your brain is converting raw data to the channel.
Yeah.
Doing continuously then?
By continuously you mean like just like you mean like all the time? Or right, yeah. I mean this is just like I think this is just kind of fundamentally how the wiring in the brain is set up. So whatever you're looking at, whether it's in focus, out of focus, like yeah, your brain's converting RGB to these coordinates. But the thing is, like color science is like incredibly complicated. The folks at Munsell, know this way better than me. Probably some people in the audience know this a lot better than me. So there's definitely not a consensus on exactly how the brain is transforming RGB to perceived color under all circumstances. A lot of it depends, like contextual, depending on like the surrounding color and things like that. But to a first approximation, that's what the brain's doing. At least that's our understanding in the field. So now, if people are curious about having intuition for what this blue-yellow maximizing model is doing, like my best way of describing this is that the visual system is basically trying to avoid the scenario where blue is in focus, where short wavelengths are in focus. It's avoiding having a sharp S-cone image, right? So it'll try to maximize contrast, but not at the expense of putting blue in focus, even when the stimulus is mostly comprised of blue or short wavelengths. And now this is just veering into speculative territory, but Emily and I have been speculating that this behavior might be some kind of protective strategy against myopia, short-sightedness, because the theory goes that Well, it's well known that the main cause of short-sightedness is that the eyeball grows too long. And when it grows too long, then everything's focused in front of the retina, right? And one theory is that if the visual system's actually trying to get green into focus, and instead it gets blue in focus, it might think that that it needs to grow longer to get green into focus, right? Because usually when blue is focused, that means green's out of focus. Um, and so maybe the visual system is trying to avoid this kind of situation. But this is just speculation. Um, I really can't put too much weight in what I'm saying. Um, but yeah, that kind of concludes the like kind of science part of the talk. The last like 5 minutes or so, I'm going to talk about some future directions I'd like to take. I guess I'm almost out of time. But yeah, just real quick. One thing about this project is that there's still some meat left on the bone, so to speak. The So in my task, I only recorded the end state of accommodation. So I let my subjects get the stimulus in focus before they recorded it, right? So the subjects chose when to record. Instead, I could have actually started recording the accommodation as soon as the stimulus appeared. And that way I could actually capture the evolution of the temporal dynamics, right? You remember I had these very nice continuous traces of accommodation, right? So I could have recorded how subjects get to the end state. I didn't because that fell a little bit out of the scope of the project when I started. Um, but that's something I want to that's something I want to reopen now in my lab. Um, and if I do so, I actually want to change my stimulus a little bit, make it more interesting. And the stimulus I have in mind is a stimulus that cycles between different color fringe patterns. I remember, so these uh, color fringe patterns, remember, are associated with different states of accommodation, right? If you're under accommodating, you get red in focus. If you're over accommodating, you get blue in focus. And that the visual system seems to respond to these to accommodate as though it's trying to get green in focus. Um, so what I want to do is actually have a stimulus that kind of continuously cycles back and forth between these different color fringe patterns, and uh, and measure accommodation as in real time as these changes occur, and also try to model the dynamics of the accommodative response. Um, One reason I'm quite excited about this direction is there actually is a theoretical paper that came out a few years ago from Jenny Reed's group at Newcastle University, where that paper basically showed you how you could apply a control theory framework, specifically predictive control, to try to model the dynamics of accommodation. Um, but the thing is, that paper was a theoretical paper. It didn't collect any new data. Um, it was more like showing qualitatively that the model could capture patterns in various other papers. But what I'm kind of looking forward to doing is to try to run some targeted experiments to actually collect accommodative data to specifically test the assumptions of this model and also try to estimate the values of various parameters in this model. So this would kind of be like a mixture of, like I said, I like to be both experimental and doing modeling. So that's something I'm kind of looking forward to. But in order to do this, I first need to set up my lab. And so this is a good time, I guess, to make this pitch that there are opportunities available in my lab, especially for any students who might have an optics background, especially if you're good with your hands at building stuff. And if you found any of this, what I talked about interesting, feel free to get in touch with me at any time. Um, so in terms of what I need in terms of setup, right? So here's just the schematic of my apparatus, right? I actually already have the measurement part down. So this a wavefront sensor that's sitting in my lab right now. I flew it over from Berkeley. TSA let me on the plane with it with no questions. Um, but the part I need now to build is the stimulus delivery, right? I want to have be able to simulate a display at different distances. Um, and so that's kind of the thing I need to do right now. And I think this is my last slide. With that, I just want to thank everyone who worked with me on this project. I mainly worked with Emily. Austin was he's well known in adaptive optics. So he's a real optics expert. And he taught me how to build a wavefront sensor. Um, and then Marty and Derek were both very gave me a lot of kind of pointers. And my research assistant Mohammed helped me collect a lot of data. And these are my funding sources. So let's see where I'm at in time. I'm a little past 4. Um, Hopefully I didn't keep you all don't have somewhere to run to, but yeah, I'll take any questions. Thanks for listening. Jim.
I'm going to ask a question that's going to mind imaging scientists when they listen to imaging scientists. Right? So imagine I work for Apple or Meta,
Mhm.
and I'm designing VR, AR headsets. What are your take-home messages for me? Is there any significance to what you're telling me? Do I need to change that? Based on your
So this is actually a conversation I've had a few short conversations with folks in the industry who have asked like a similar question. And I think I actually think the biggest application of this finding potentially could be more to like the myopia control literature. So there's a lot of attempts right now in industry, mostly from like pharma companies. But I know Meta and Apple are, especially Meta, are pretty interested in this question. Like, how can we stop the development of myopia? Because kids these days are getting, like like astronomically high rates of myopia. Theory being that spending a lot of time indoors. Um, and so some theories of how myopia develops, like there's no generally accepted theory, but some theories hold that it's actually the whether or not the eyeball decides to grow longer, causing myopia, um, could be a response to the specific wavelength that's in focus on the retina. So I talked about the the visual system might be trying to avoid blue in focus. So maybe this is suggesting that you might want to have displays that try to present stimuli that keep you away from having blue in focus. Um, I don't know exactly what Meta and Apple and Google are thinking in terms of myopia control, right? That's something they won't tell me, but I know in like the contact lens world, right? There have been some proposals that you have little colored that you have little maybe not contact lenses, but like color filter glasses, or for screens that kids are using, you apply color filters to the imagery to try to put certain wavelengths in focus that would be advantageous to preventing myopia. But what those wavelengths are, still up to debate. The animal literature is like extremely mixed on what wavelengths might cause myopia. So this is kind of where my work kind of intersects, kind of industry interests. Yes.
Back to few slides back, like when you model when green and red models for the LMS. Like I was wondering if the number of cones like for these like LMS like could be weighted during like the prediction of luminance.
That's a good question actually. So it's widely believed in the color literature that like S cones like either don't or contribute minimally to luminance. It's mostly L and M. But you also get, people have run color experiments have also kind of shown that like the weighting of how you combine L and M varies from individual to individual. So that's actually why I made it a free parameter in my model.
Yeah, because of like just simple plus and minus. There's no weighting factor.
Right. Yeah. So actually, the W in here that's the weighting. Yeah. This guy's kind of hiding in this gigantic plot. Other questions?
Are there any other questions?
Happy also talk more offline if anyone's interested.
So let's thank our speaker again, and if you have any questions.
Thank you all.